Welcome, everyone. On behalf of the Center for Brain Health, welcome to our Frontiers in Brain Health. This is where we take a deep dive into some of the most exciting new developments in brain research. My name is Dr. Lori Cook, and I serve as the Director of Clinical Research here at the Center for Brain Health. And yes, you can think of this as our winter season finale. So we are so glad that you are here today. Just a couple of quick housekeeping comments. We will have time at the end that we'll reserve for some time for questions, including both those who are attending in person as well as those of you who are joining us online. So for our virtual audience, please utilize the Q&A function to submit those questions. And those of you here, just keep record of your questions that you'll be ready to ask at the end, and we'll give you that opportunity. Another quick reminder, this presentation is being recorded. So quickly, for those of you who may be new and are not familiar with us, the Center for Brain Health is a cognitive neuroscience research center for the University of Texas at Dallas. And for the past 3 decades, we have been leading the scientific quest to measure and improve brain health across the lifespan. And from our perspective, the bottom line is that optimal brain performance is the new frontier of human potential. And so we're so excited to welcome speakers who can really help expand our knowledge and curiosity around some of these topics. And today is absolutely going to be another one of those to expand your mind. Please allow me to give a quick introduction to our speaker today, Dr. Stacie Warren. She is one of our illustrious colleagues here at the University of Texas at Dallas. She's an associate professor who directs the Computational Neuropsychology and Affective Neuroscience Lab. I love she characterizes herself not only as a skilled clinical psychologist, but also a lifespan neuropsychologist. So we'll get to see what that looks like today. And her lab's work focuses on the etiology as well as the progression of psychopathology. So looking at both internalizing and externalizing disorders. And what I find particularly interesting is she's really trying to help us better understand what are the shared mechanisms that help explain why these types of conditions so often co-occur. So she's using a lot of different tools, neuropsychological assessment, laboratory-based tasks, self-report. She was sharing about doing some really cool clinical interviews online when people are in their homes, and even some sophisticated computational approaches to help build these more precise and even inclusive models of mental health. So, excited for her lecture today, and will you all please help me welcome Dr. Stacie Warren.
All right, how's that sound? Okay, so I am coming down from losing my voice. So at some points during today, my voice might be a little cracky or it might sound very, very deep. Just go with it, okay? We'll see how long this lasts. All right. 2 things at once. So these are some members, but not all, of my lab. A lot of folks that I collaborate with. Okay. So today I'm going to be talking to you a bit about my research and my team's research, and basically focusing on how we can look at cognition, or more specifically, cognitive control, to try to understand things like anxiety and depression. So a lot of my work focuses on things like stress, adversity, symptom dimensions, and how these things might affect and shape our brain, as well as taking a look at how computational models might be able to help us understand these things, all right? So today you're going to hear a lot about anxiety and depression, a little bit of sprinkling of externalizing stuff, but mainly anxiety and depression. All right, so what we are seeing is that anxiety and depression are on the rise in kids and adults. And this actually happened before COVID-19. We like to blame the pandemic, but the reality is that this stuff started long before that. And there's just something in our world that is making mental health a little more challenging. All right, so what is really puzzling is that our treatments have not improved substantially in the last 50 years. Even though we have a lot more tools than what we used to, response rates are not where we'd like them to be, with some folks experiencing limited or no relief. And relapse is very common. So if you have depression, more than likely you're going to experience another depressive episode. And we continue to have what is this comorbidity or co-occurrence reality. So anxiety and depression like to co-occur. They like to occur together. So the fact is that our methods aren't as effective as we would like. And this tells us that we're missing something fundamental about how these conditions develop and maintain themselves. So most mental health problems have their origins earlier in development, and the vast majority of mental health problems emerge before the age of 25. So if you think about it, this is a key time in the development of our brains, all right? Our brains are rapidly developing in our youth and emerging adulthood. And so when you think about treatment, okay, our treatments are usually centered on a diagnostic label, all right? Maybe a question then, when we take a look at these developmental spans, maybe a question could be, should we be tailoring our mental health treatments to the stages of our brain and behavioral development, all right? So a recent study seems to suggest yes. So this is a study of the effectiveness of cognitive behavioral therapy that just came out. This is using participants from the UK. And what we see here, can I walk? Okay. So what you're seeing here, these are change scores from pre to post treatment. So the bigger the number, the bigger the change, all right? So what we're seeing in terms of the cognitive behavioral therapy effectiveness is it looks good mostly, except when we start around age 15-ish, up until about the age of 30. We see a dip in both of these lines. So what this is showing us is that cognitive behavioral therapy isn't as effective as it is for individuals who are older, all right? Now, this is striking because, again, this is when our brains are more adaptable. So if our therapy isn't working at a time in which our brains are the most plastic, the most adaptable, then that's a major clue that we might be missing something. So what are we missing? I'm going to focus on difficulty concentrating. So this is a symptom that is shared across anxiety and depression, as well as other forms of psychopathology. So what do I mean by this? Imagine that you are trying to work on something and you just have all of these worries that just keep coming in and you're not able to do the task at hand. Or perhaps you're just so unmotivated that it's really difficult to make a decision. In fact, what should be a simple decision requires a lot of effort, okay? This kind of stuff, these are real cognitive struggles of anxiety and depression, and yet in the therapy room, we really don't assess that carefully, okay? So why does that matter? Our gold standard treatments are described as cognitive behavioral, but yet there's elements of cognition that we do pay attention to in therapy, but there's a lot that we do not assess carefully. So In fact, anxiety and depression, there's a main target is irrational thinking, irrational thoughts. Sure. But beyond that, anxiety and depression have some cognitive symptoms. So that mental fog, difficulty concentrating, difficulty making decisions, okay? Those types of symptoms have a tendency to hang around after treatment is completed. So maybe then irrational thoughts, irrational beliefs, there's something more to that. What if the problem isn't just the irrational beliefs, but how your brain is actually processing information? All right, so I put eyes on a brain because I wasn't sure how people were oriented to brains. So here's the four. Okay, the forefront. Okay. All right. So anyhow, information processing. So information processing, what do I mean? Well, information processing, like how we take in information into the world, that affects something called emotion regulation. So emotion regulation, put simply, is your ability to change how you feel, all right? Now, when we regulate, all right, this involves two broad systems. So it's a cognitive top-down system and a bottom affective system, all right? So these systems work together. But how these systems become compromised in the development and emergence of psychopathology is just really open territory. We don't really know a whole lot of that. The other element of this is that we don't have a good sense of cognition in the emotion regulation process, all right? So understanding how these systems become compromised and for whom, under what conditions, these things are essentially relevant to mental health. So how do we test emotion regulation? So what I'd like you to do is just look at the picture and just describe how the picture affects you. Just take it in. If you saw that in front of your face, how would you feel? Scared? Terrified. Why? Yeah. So this is not a friendly looking Doberman, right? It looks like it's going to come after you. But now if I said, all right, I'd like you to think about this image in a less negative way. How might you think about it? If I'm asking you to think of this image in a less negative way, how would you interpret this? Happy to see me. So one way that you could look at this as well is maybe that's my dog and he's protecting me. Maybe there's something behind me, right? Now, do we see the differences in how that made us feel? Okay, the first time around it's like, oh, danger, okay? The second time around though, it's like, oh, protection. He's my friend, right? This is something called cognitive reappraisal. As a therapist, this is what we teach. We teach some of our clients. This is a tool that we use to teach our clients. It's also a tool as a researcher that we love to use because it's a way in which we can tease apart those top-down and bottom-up systems that I just talked to you about, all right? So I'm going to walk you through a study. Okay, so this is a study in which we were looking at how symptoms of anxiety and stress reactivity affect emotion regulation in kids. So they were given that same reappraisal task that you just did, okay? So they were asked to look at pictures, rate how they felt, and then on the reappraisal portion, they were asked to think of the image in a less negative way, and then also rate how they felt, okay? Now they're doing all this while they're in the magnet, so we're scanning them. These kids are unique in the sense that they're coming from a highly trauma-impacted community. So these are kids that were all living below the poverty level, all right? And remember, we're interested in this paradigm because we want to tease apart those top-down and bottom-up systems. So I'm going to walk through just some brief hypotheses. Okay, so for the top-down and bottom-up systems, there's a particular pathway that I'm focusing on. There's a lot of brain regions involved, but I'm going to focus on two in particular. I'm going to focus on amygdala. I'm sure everyone's heard of amygdala, because when you hear emotion and brain, everyone's like, amygdala. But I promise you, emotion has a lot more than amygdala that goes into it. Okay, so amygdala is part of that bottom-up system. And then dorsolateral prefrontal cortex, or DLPFC, that is part of that top-down cognitive system, okay? So there's a pathway, and it talks to each other. Now, in terms of high anxiety, high stress reactivity, we anticipated that we're going to see a directed effect between amygdala and to the dorsolateral prefrontal cortex, okay? And then that would be indicative of maladaptive emotion regulation. Then we also hypothesized the reverse. So the directed effect between DLPFC and to amygdala, that would be indicative of adaptive emotion regulation. All right, so what do we find? Okay, so what we see is that the intervention was effective. The kids rated the reappraised images as less unpleasant compared to the aversive condition, so taking it in as they normally would. Then we looked at the behavioral data. So with the behavioral data, rather than looking at reaction time, which is a common thing to do, we decided to use a computational model, something called drift diffusion modeling. And the reason for that is because drift diffusion modeling can actually pin down very specific elements of the reappraisal process that reaction time generally isn't as sensitive to, all right? I'll come back to that in a few slides, but just hold that for a minute. So I'm going to talk to you about the different drift diffusion modeling parameters and what they mean, okay? All right, so what do we see behaviorally? This is an illustration of a single trial of the drift diffusion process, and I'm going to walk you through it. So this oval, that highlights an individual's bias or an individual's predisposition to positive or negative ratings during the reappraisal process, okay? So for children who were reporting high anxiety, their initial bias was more negative from the start. This means that they required more effective cognitive capabilities in order to kind of overcome that negative bias. But what we found was that they had a slower, what we call, this is called a drift rate. So this is how, like when you're processing information, how fast that you can work with that information. And kids with high anxiety, this is kind of low and slow, right? Low and slow. And then lastly, what we found is that kids with high anxiety and high stress reactivity were taking less time to make a decision, as evidenced by what we call a reduced decision threshold, okay? So if you have a small decision threshold, that means you're only taking a little bit of information before you push that button. Bigger decision threshold, you're taking it all in before you make that decision, all right? So to kind of sum up here, what we're seeing is that high anxiety seems to put kids at a disadvantage when trying to reappraise aversive images. From the get-go, they're starting off in a negative place. The reappraisal process itself isn't as effective, and for whatever reason, high anxiety and high stress reactivity, it's either reducing their capacity to take in more information, or for whatever reason, they're just not taking in as much information before they push that button. What do we see in the brain? Okay, what do we see in the brain? Maybe this one. There we go, okay. All right, so remember we're interested in teasing apart that bottom-up and top-down pathway. And I focused on two regions. So this is the amygdala and the dorsolateral prefrontal cortex. So what we did find is that kids with high anxiety and high stress reactivity, we saw a directed effect from amygdala to DLPFC. So that bottom up was present, right? But when we tested the reverse, so DLPFC down to amygdala, we didn't find anything. So to us, this is really important because this pathway has been theorized, many different theories, as being very, very important for emotion regulation. What we would typically find in effective emotion regulation is that directed DLPFC down to amygdala effect. And here we didn't find that. So what does this mean? At least for this sample of kids coming from a highly trauma-impacted area, it seems that anxiety and stress reactivity kind of hijack our cognitive control, so our ability to be able to really regulate our emotion. Okay, so remember when I said, hey, we're going to use this really complicated model to look at our behavioral data. Why did you do that? So we chose to do that, again, because that kind of parses down the decision-making process during emotion regulation into specific elements. But when we did look at reaction time, what we found was that anxiety and stress reactivity in the sample did not significantly correlate with reaction time. So everything that I just showed you would have been missed if we did not use this model. Now, I don't want to come across as indicating that using reaction time is wrong. It's not wrong. It's just for us and for our purposes and for what we're trying to target, computational models, they're mechanistic, whereas reaction time is descriptive, all right? So in fact, what my lab has been doing is we've been evaluating how computational models and approaches might be able to help us better understand cognitive disruptions across psychopathology. So this is a study in which we took, So this work is pulled from a very large sample size, the Adolescent Brain Cognitive Development Project. So there are over 10,000 kids in here. And what we did is we looked at flanker task performance, all right? So this is another cognitive control task. We compared traditional reaction time measures from that flanker task to a diffusion model or a computational model. And we looked to see how well each performed at predicting psychopathology symptoms, okay? So we've got anxiety, depression kind of stuff, we've got attention problems, rule breaking, everything, all right? And what we found was that the drift diffusion model, the computational model in orange here, that outperformed the typical reaction time models in purple here, across the board. But more importantly, what the computational model gave us is it showed us that across all these very different symptoms, they all shared the same thing. They all shared a reduction in their drift rate or how people are processing information. It all showed a decrement, all right? Then it also showed us something very specific. It showed us that there was a reduction in non-decision time, and that was specific to rule breaking and aggressive behaviors. So what is non-decision time? Non-decision time is your brain getting ready, just preparing to do the task at hand, okay? So if you have a reduction in non-decision time, in the context of rule breaking and aggressive behaviors, we interpreted that to mean that these individuals probably were more impulsive. That's how we interpret that. Okay, so Now what? All right, so this is a project where we're trying to take our computational models and align them with some of our brain data. So this is a project, it's very, very new. These are results that we're still chewing on. And this project is being led by my postdoc, Ritesh Malaya. And so I'm gonna walk you through the cognitive control tasks that we're using and then the model, all right? Okay, so in the same adolescent brain cognitive development group, so this is over 3,000 youth, they performed what's called the stop signal task. So we focus on the stop signal task because it has a long history of theory in terms of, how people withhold a response, okay? Withhold a response. So for those of you who are not familiar with stop signal task, usually what happens is you'll see a cue, a go cue. In this case, it's green arrow, so you just keep pushing that button, okay? Then you'll see a stop signal. In this case, it's a red X, and you withhold. The stop signals, you don't know when you're coming. And sometimes they come super duper fast, and sometimes they come a little bit later, all right? So at various points, this task can get quite difficult in terms of withholding that response, all right? So now, what we're showing, this is a typical stop signal task reaction time measure that we use in research. So this is traditional. When we look at children who they score low, so this is typically developing kids, they're all low in these symptoms. That's a pretty uniform profile, okay? It looks very similar across the board. But now when we add kids who are scoring high in these symptom domains, we start to see a lot of variability, okay? We don't know why they're variable though. So to approach this, we are taking a kind of a two-step approach. So the first thing is that we applied a computational or process model to capture this variability. And the second part is that these kids, while they were doing the task, they were in the magnet. So we took their brain data and we extracted data from regions of interest that we know are involved in cognitive control, all right? So now we've got process model, we've got brain data, and we want to put them in a similar space so that we can compare them, because we want to see if the variability the process model is capturing, does that match the variability in the brain data, all right? So we did something called representational similarity analysis. It's basically a geometric representation of these things. And it's like a grid. And you can put them together and determine, are these things similar? Are these things different? Okay? What did we find? Okay, I'm going to walk you through this. So first off, the bars. Ignore the lines. These are the bars, okay? These are the youth that scored high on the symptom measures, and what we're doing is we're comparing their typical standard reaction time performance. This is the difference between their performance and the typically developing kids. So the blue bars mean that some of these youth who scored high, they were faster on the stop signal task. The pink represents the youth that, again, scored high, but actually performed more slowly on the task relative to typically developing youth. Okay, so now these lines, okay? These lines represent our behavioral process model and then also our brain model. And the reason that we want to look at them is because if they actually match up, that seems to indicate, well, we got something. And also, that might actually explain some sort of shared mechanism. And here, what you see between the imaging process representation and the behavioral process representation, these things are aligning quite well with the empirical data, really well. It's not perfect, but it's pretty darn good, So the brain data here is collapsed across all the ROIs that we sampled. So what if we look at specific ROIs? So the top panel represents the youth that, again, they have symptoms, but they actually performed faster on the task. And you can see here that these four regions of interest, these are the ROIs that are working together for them to be able to do the task. The panel on the bottom, these are the youth that performed more slowly, and these are their ROIs that are helping them perform the task. You can see that there's some similarities and also some differences, but this is the one thing that I want to point out. This blue spot, that is dorsolateral prefrontal cortex. That's the same region that I talked to you about during that reappraisal task with the dog. What does this mean? So all of the pinkish reddish regions mean that the brain data is in alignment with the process model. What is not in alignment with the process model is DLPFC. These things are not similar. So DLPFC is doing something, and the process model is doing something. They're not in alignment. What if we took a broad categorization rather than looking at very specific symptom clusters? So this is something that we do. So internalizing and externalizing. This is a common way to look at psychopathology. So remember, internalizing, this is all of your anxiety and mood stuff. And externalizing, this is all the rule-breaking behaviors, your aggression and whatnot. So let's talk about internalizing first. So what we see, so this is again for the kids that actually performed faster on the task relative to typically developing children. There's a big engagement with posterior cingulate cortex, left frontal eye fields, and a relatively lower anterior insula. This is, we're still chewing on this, but this seems to be a pattern that actually is common with someone who's really engaged, like overly engaged, maybe ready for that stop signal to come out at any time. Externalizing, so I want you to notice something. We have a direct, opposing pattern here. So in terms of externalizing, we have a big engagement of anterior insula and a relatively lower posterior cingulate cortex engagement and no frontal eye fields. What does this mean? We're still chewing on it, but anterior insula, so anterior insula is a hub of what we call the salience network. It does a lot of stuff. But one of the things that it does is it says, hey, pay attention to me out here. Look at that. Oh, there's something in your environment that you need to pay attention to. It's also called like a circuit breaker. So if you need to break away from whatever it is that you're working on to pay attention to something in your environment, that is what anterior insula does. This kind of presentation seems to indicate more of a saliency kind of networky, maybe less regulated pattern, which kind of makes sense if we think about externalizing being externalizing disorders are having a lot of what we call problems with inhibition, okay? So this is actually a very interesting finding from our perspective, but again, this is all new, so we're still chewing on it. We're still chewing on it. But this is really fascinating work, and Ritesh did it. Okay. All right, so now, I've been talking to you about symptoms, all right? I've been talking about symptoms, I've been talking to you about brain regions, but one thing I have not been talking to you about is a broader context. So let's talk about context. What do I mean by context? We as individuals don't live in a vacuum, all right? We have a variety of social, cultural, environmental factors that shape us. And so this is a project that I've been working on with Dr. Norit Chandra and his student Farabi Shuvo. They're also here at UTD. This is a project that came out of developing a model, a computational model for cognitive control. So with this model, this new model that we developed, this is an application of that new model, all right? So just hold the model in your head just for a minute. I'm going to talk to you now, who is this sample? So this, again, is ABCD folks. There's about over 8,000. And there were over 22 measures of someone's social environment. So these are things like SES, perinatal health care, social relationships, social support. So it's not just a simple, what's your SES? This is a whole slew of measures, and there were latent factors, and we took them, and then we said, okay, out of all these latent factors, could we cluster these and see, do we have any sort of profiles that might emerge? And it turns out we do. We've got 5. So we have the advantaged overall group, so that's everyone was great on everything, and then it kind of goes on down the line to where we have the highly vulnerable group. So that means low SES, limited social support networks, limited perinatal health care, so on and so on, all right? Then when we took those vulnerability profiles, we overlaid who is anxious. And as you can see, in the highly vulnerable profile, we have a larger proportion of kids that are anxious, especially relative to the advantaged overall. So I'm just going to focus on these two to compare them. So now let's go to our model output. Okay. So I'm walking over here first because I'm going to look at the advantaged group. So the red line, this is going to be counterintuitive. The red line is actually kids who are not anxious, okay? The green line are kids who are anxious. So for this model, we looked at some of the things that you've already heard about. So drift rate, right? So how quickly they can process information, and then also how much information they need before making a decision. What we find, and what you can see, is that the typically developing kids, those who are not anxious, those perform better relative to the anxious kids, okay, within the advantaged overall group. Now when we look at the highly vulnerable group, what you see is that there's no longer a distinction, okay? There's no longer a distinction between anxious and non-anxious kids. They kind of start to align, okay? They start to align. And you could kind of argue that within this group, the being highly anxious, at least when it comes to your ability to process information, there's a little bit more variability in that. But overall, by and large, being anxious, it seems to kind of close that gap, all right, within the highly vulnerable group. So overall, the highly vulnerable group, no matter if you have anxiety or not, they are not going to perform as well as the advantaged group. But we wanted to see if we could actually tease that apart a little bit more, all right? So anxiety does seem to play a role in here, but let's test this more specifically. All right, so with the model, what we did, or what Farabi did, was he took all of the model, the computational model elements, and then he put them together. And he ended up getting 3 major components, or 3 cognitive profiles, all right? So we've got one cognitive profile that is the most vulnerable profile, which means that group of individuals, for all of the parameters that we were looking at, they were low. Then we have an average cognitive profile, and then we have a really strong cognitive profile. Then we overlaid well, who makes up these cognitive profiles? Who's in them? So we overlaid our vulnerability profiles to kind of see who fits where. So you'll notice in our most disadvantaged cognitive profile, we also see that the folks that make this up or have the most membership are the ones that are in the most highly vulnerable of the socioeconomic, social environmental groups. So the pink and the blue, which I didn't show you, but they actually look very, very similar in terms of their cognitive performance. This is what makes up that highly vulnerable cognitive profile. In terms of the strong cognitive profile, that is largely made up of individuals who are in the advantaged and socially resilient, which actually looks very similar to each other. That's who is in this group. So what does that tell us? That tells us that our environment, our background, really plays a significant role in terms of shaping our cognitive abilities. What happens when we look at anxiety? So I still have these same cognitive profiles, 1, 2, and 3 over here, and now I'm looking at anxiety membership, okay? I'm looking at anxiety membership. And what we can see is that when we look at the very strong cognitive profile, anxious folks are a little less represented, but not by a whole lot. Okay, not by a whole lot. So really, when we started testing this more explicitly, we find that anxiety, it kind of explains a little bit in terms of someone's cognitive performance, but really, in this sample, a lot of the explanation, the big weight, if you will, is more about these social environmental factors. Okay. So, what can we conclude? Ooh, is it 12:45? Well, we're not going to get to adults today. All right, so I'm just going to leave it here. So, what can we conclude about all this? Cognitive control is a key mechanism that's involved in emotion regulation and the development of depression, anxiety, and it definitely seems other symptoms. So what that means is we can call cognitive control or decrements in cognitive control as a transdiagnostic risk factor. So that means what we saw was the same problem with cognitive control, and it wasn't just about anxiety and depression. It was anxiety, depression, and externalizing phenomena. So that's what makes it transdiagnostic. We also see that social environmental risk factors play a huge role in cognitive abilities. What I didn't get to today are some gender differences on that front, but there likely are. And another study that I haven't shown you today in terms of gender differences in the developmental trajectories of the symptoms themselves. That's also playing a role here. In terms of that amygdala to DLPFC, that pathway, it's an interesting question of, well, gosh, could this then be some sort of a transdiagnostic circuit? Meaning that we saw that it's playing a role in anxiety and stress reactivity, and then we saw in another study a reduction, like DLPFC was not playing the same in terms of our cognitive model. Perhaps this could be a transdiagnostic circuit, and more specifically, we can talk about or think about DLPFC in particular. So again, part of that top-down regulatory control. Maybe DLPFC is some sort of node of vulnerability, because there seems to be a problem with it. And in fact, in adult work, we have something called TMS, where we kind of, this is going to sound bad, but zap folks just a little bit, kind of give their brain a little bit of a rev. This is what we target for folks who are depressed. We target DLPFC. And there's some newer research that's actually doing the same thing, but on the other side of the brain for anxiety in adults. All right. So what does this mean then in general? Like, why am I doing all this stuff? So just a minute, I'm going to fast forward to a lot of slides. Okay. Why do this? So the idea is that if there's something more to anxiety and depression, and clearly there is, maybe that's a reason, so people's cognitive control abilities, maybe that's a reason why some of our treatments have just kind of stalled. So if we can actually take that and put that in the therapy room, I'm wondering if that might improve our treatment effectiveness. So before I came to UTD, I got here in 2022, we started actually doing what's called a transdiagnostic treatment. So meaning we took a kind of cognitive behavioral therapy treatment and we gave it to everybody. This is actually a pilot that was done with a local homeless shelter. And what we were attempting to do is test people's cognitive control abilities before we gave them the intervention. Because we wanted to see, could we then predict how effective the intervention might be based upon their cognitive abilities, okay? So we started that, we piloted it, and then then COVID hit, and then I came here. So I would eventually like to get back to this at UTD, but we're not quite there yet. Okay, so ultimately then, the goal of my work is to try to improve treatment effectiveness. That includes approaching research in anxiety and depression, among other things, from a different perspective. And that different perspective is focusing a lot on cognitive control and leveraging computational models. That also raises a question in terms of lots of theories of anxiety and depression, because a lot of that stuff was also built when we weren't applying computational models, all right? So really what I'd like to do is be able to personalize treatment, to personalize it, to hopefully improve effectiveness. Because at this stage of the game, even when it comes to medication or psychological intervention, we're essentially taking, it's a guessing game. A given person walks in, and you're trying to figure out, is this intervention going to work? Is this medication going to work? And you kind of go through cycles of that. And we all know, or clinicians know, that as you're going through cycles of that, you risk losing those patients because they're like, why am I doing this? This is not working. So if from the get-go, we can do something different to try to better match a client with the type of treatment that would be best for them, that is what I'm hoping that we can bring to more of this personalized medicine approach. Okay, I think I'll end there. Thank you.
Thank you so much for that. And I have no doubt we have several people who are interested in asking some questions here in the audience. We'll start here. Happy to run the microphone around. Thank you, Bonnie. And then those of you in the online audience, put in your questions. We'll save some time for y'all as well.
Okay. Thank you, Dr. Warren. I really appreciate your insight on this. I have a question about the CC3, so the cognitive control groups or the clusters. I thought it was really interesting to see all 5 of the populations throughout all 3 of the clusters, first and foremost.
Yeah.
Were there any common themes or observations that the team found about that highly vulnerable population who still managed to be in that strong cognitive cluster?
Ooh, that is a great question. So that's also new. I don't think we've dug into it just yet. So, okay. So the short answer is no, but we haven't really dug in deep. What I will say, though, is we're painting, these are on average, right? These are on average. So there's other things that are represented in those clusters that we did not explicitly measure, right? So an interesting thing about trauma, adversity, and whatnot is that there are some individuals, for whatever reason, their path leads them to things like anxiety, depression, and depending on age, externalizing disorders and whatnot, right? And then there are some that are resilient. I don't know what that is, but I would love to be able to understand that and capture that. So what I will say is the short answer is no, because we haven't looked deep into it yet, okay? But what I will say from a developmental perspective, from a trauma perspective, not all paths are created equal, and just because you do have, like if you come from, a poor background, you come from a lot of adversity, it doesn't necessarily mean that's your path, like that you will end up with anxiety and depression. It's not uncommon that you would, but some folks, for whatever reasons, they're resilient. And I don't know what that is.
Awesome, thank you.
Thank you so much for this topic. It's so timely in our days and our lives now.
Sure.
As a speech-language pathologist, this has been part of my world for nearly 50 years, and so thank you for, again, when you were testing the cognitive abilities, could you speak for a moment about what you did to make that happen? I used to get in some knockdown, drag-out fights with some other professionals in the schools, particularly with what is a cognitive ability versus a child who also had language deprivation or language disorders, auditory processing disorders, and how we need to separate that to get a true cognitive ability which sees someone learning in a very different way.
So I am a neuropsychologist. So I've had my fair share of testing, and I'm a lifespan neuropsychologist. And for the longest time, evaluating learning disabilities was my bread and butter. So what I will say in terms of, testing, the groups that I've used here, with the exception of the very first study, the big sample sizes like the 8,000, 10,000, 3,000, those are not mine. Those are from the Adolescent Brain Cognitive Development Project, but in terms of testing, everyone got the same. And for that particular data set, it is a national study. So there's 21 sites across the United States, and they purposely tried to oversample kids who are underrepresented in research. So from that perspective, they attempted to do that. Now, in terms of what you're asking, I think, is how do we separate folks who may approach a problem differently relative to maybe other kids. So a lot of variability, a lot of diversity in terms of cognition. As a neuropsychologist, I would say something that I do is I'm more like process oriented. So I'm kind of like, I don't do the one size fits all in terms of testing. There's things that I look for when I'm evaluating a child or an adult in terms of how they're actually approaching each measure. When you're asking, well, what about the tests themselves? That is a whole other topic that I have lots of lectures on that I can't get into today. But what I would say is a lot of our standard clinical tests, okay, let me say it this way. There's a reason that I started using computational models. And that reason is because as a neuropsychologist and as a researcher, I was always frustrated by our standard clinical measures as not giving us what we wanted, in the sense that they weren't the best predictors of everyday real world behavior, okay? That doesn't mean that they're not useful, so I want to throw that out there. It doesn't mean that they're not useful. It's just trying to translate how you performed on the Wisconsin card sorting test to, can this person drive? Two different things, two very different things, all right? So the reason that I started pivoting to computational models was, one, I felt that the metrics that were used for these standard clinical tests were not giving us what we needed, nothing specific. And the other is to start to see, well, from these clinical tests that have been around for a long time and just revised and prepackaged into something else that they call it something else, but it's really the same thing, could we gain something from that using computational models? And then the other approach I've taken that I didn't show you today is I've actually started developing some of my own tests in the lab. Yeah. So I hope that kind of sort of gets at your question.
What can we do more is the bottom line.
What can we do more? Yeah. I think it's going to take a radical shift. So neuropsychology, I'm going to pick on neuropsychology, and I can because I am a neuropsychologist. But in terms of as a field, as a profession, we are behind the times. A lot of us still use standard paper, pencil measures, and the rest of the world is using things like AI. And I think neuropsychology is ripe for a paradigm shift, not just in terms of how they approach assessment, but also the assessment instruments themselves. Yeah.
Stacie, amazingly
Sorry.
elegant research. Thank you for presenting and being here today. My question to you is, what is your hope if we take the most vulnerable and if we have a cognitive training program that actually helps to develop this kind of dorsolateral prefrontal cortex to downregulate the emotional, what is your hope that this could make a difference for the most vulnerable in terms of outcomes, regardless of where they are and started?
Yeah, so great question. So part of the reason I also got into this is because I was very much interested in cognitive training. And the cognitive training research that's out there, there's some mixed results in terms of the effectiveness, but there's also, because of those mixed results, there's this, wait a second, maybe if we kind of zero in on the signal, maybe we can make it much more effective, okay? So in terms of cognitive training, what are my thoughts on the most vulnerable? I think that if cognitive training were done, but cognitive training typically has a tendency to take standard tests that we use for like one-shot assessments in clinical land, and then trying to change the stimuli just a little bit and go, oh, hey, keep doing this over and over again. That's not really representative of the world either, right? So I think what we would have to do is we'd have to like step away from all of the standard clinical stuff, develop something new that in terms of clinical training, and then implement that. And I would hope that in terms of those who are most vulnerable, that might be able to help them more effectively regulate, because we might help them introduce some flexibility in their thinking patterns. So I didn't get to that today, but a lot of cognitive flexibility is what I'm hoping for.
Wonderful. Well, I think we are out of time for today. Thank you so much. I think we see a lot of common ground here. Appreciate you sharing today. And interested to explore ways to partner as we look through the lens of cognitive training, cognitive strategies, and also large-scale data and how we can use that to not only better understand the mechanism, but also better inform more effective, personalized approaches to brain health. So thank you. And thank you all today. Again, this was our winter season finale for our Frontiers of Brain Health. So we hope that you will be back to join us again in January. January 16 will be our next lecture. And in the meantime, all the best to everyone here. I hope you all have a happy holiday and a brain healthy new year. Thank you so much.